We continue with 8.2.2, but this time we're going to work with microscopes, micrographs, and of course magnification. Now, what is this all about? It's about you learning how to use microscope in class and be able to measure whatever specimens that you're looking through the microscope and be able to draw them. Now, before anything, you know that the subtopics always have a guiding question, so we are going to keep those in mind all times. That's why it's again here. Now, what are the things you're going to learn? Of course, you're going to learn how to use a microscope, so you have to get the skills, how to actually create the mouthpieces. You're going to be able to look at it, how to actually use the microscope and which pieces you're moving or what. You're also going to look at different types of microscopes that have been developed throughout the years. You're also going to look at the cell, different types of cells, and you're going to look at their structures and see and be able to recognize them in the micrographs. And eventually you're going to have to know how to draw them and annotate whenever you're looking at those pictures and be able to point them out and label them out. So with that in mind, what are you going to see in this video? You're going to actually learn everything from how staining the mountains. You're going to understand what the eyepiece, Graticule, and stage micrometers, what they're measuring. You're going to do some calculations with magnifications, image size, and of course, actual size. You're going to see some applications of electron microscope, as well as the techniques that are commonly used in a microscope. So to start, why not with getting you exposed to something that you probably see every time in high school, which is the light microscope. Now, light microscope is basically using the light and lenses in order to see things, and it's found that easily in your school. Now, the electron microscope, this one is using electron beams, and these guys are not found in schools. It's impossible because they're very expensive, unless your school is very rich. But anyways, these ones are usually found in universities or the big labs. Now, if we zoom in like an X-ray, you will see here that they are looking differently because they function differently. We actually have on one side, which is the light microscope, using lenses and light in order to be able to see the process. On the other one, you notice that there is some source of electrons and there's an electron detector that is picking up these electrons. So they work differently in how you get to see these images. However, since we're getting to this, we should know what are the differences between them. So the light microscope is extremely inexpensive. That means you will find about 15, 20 of them in your school. While you will see that when it comes to microscope, electron microscope, they're very expensive. When it comes to light microscope, they're so easy to prepare, while the electrons are very complex and very hard to put them in. So it takes time to get them right. Next, light microscope, it's 2,000 times. It makes the specimen looks 2 times bigger for you to see it. However, electron, it's about 500,000 times more bigger. When it comes to light microscope, you can see any specimen in living or dead. But when it comes to electron, it's completely dead. You cannot see things living, which is a sad thing. So these are the comparisons that you might come up as a question. Might be like compare those two, contrast those two or distinguish those two. Now, when we're looking at pictures, obviously you're going to see that the electron one is more detailed, as you see here. You see how much detail that is? Because that's what the electron microscope does. It goes extremely detailed, so the magnification is really big. However, in the light microscope, you can see the specimen, but you see it blurry. This is the reason why, once again, the electron is really good for you if you want to see the details, and the light microscope is even better because you still can see something living. So with that in mind, we're going to look at some other different types of microscopes being used in order to see the pictures or whatever of the specimens. So here we have the freeze fracture. It's basically where you freeze actually the specimen and then you break into pieces. And then you put them into an electron microscope and you just visualize what is there. And usually you can see the tiny little internal structures inside of the cell. So you can see right here how detailed these are. The second one is cryogenic electron microscopy, which this is you also freezing the sample, but now using cryogenic temperatures. They are fixed, firm, and stable. And these guys, obviously using electron microscope, you can see the image more in detail. And at the same time, is reducing any kind of damage that is going on because of the cryptid. So this is what usually the picture looks like. Then you have the third one, which is immunofluorescence. This one is very much used, and sometimes you see those nice little pictures in your books of images of cells and stuff, just like this one right here, and that's what it is about. It's about immunofluorescence. So there is a type of fluorescent tag known as fluorophore. Now this is attached usually to an antibody. Now what is an antibody? Antibody is a protein. It's a piece of protein here, and that antibody will always attach to an antigen. Both of them are proteins. So they have to match. They have to somehow connect. The antibody and the antigen, they have to connect. Now, using this pencil here, let's say that this is the fluorophore. So the fluorophore attaches to the antibody, and when they attach, clicked, good. But they have to be specific to each other. And then somehow you have a light hitting on that tag, which is the fluorescence. And that is where these colors are going to project from it. However, there's a lot of specimens. If the antibody doesn't touch to anything, that means you're not going to see any colorful pictures in there. So this is what this whole process of this immunofluorescence looks. So in the end, a wave of light is just going to be hitting on that tag, and then you're going to see different colors coming out and looking bright like this picture. Another way also of colorful pictures is fluorescent dyes. So this one you use a light microscope, and it's easily when you just drop a dye into the specimen, and then the bright color will show up when the light hits on it, as you can see right here. So these were the 4 different ways of using microscope in order to see pictures. But there's one thing that students really don't understand, is that you still need to know how to use those words, resolution and magnification, which I find it very different because you guys have iPhones in there, and always they say, oh, better resolution. What does that mean? It's just that the pictures are more detailed. And magnification, you're making the pictures bigger so you can zoom in. That's what it's all about. So every time my phone gets better is because it's saying you can zoom in more, that is the point of magnification, make it bigger, and resolution, which makes it look more detailed. That's what it means. So it's the same thing with a microscope. Now, some students still also cannot get the scope of what are the sizes, why we have to use a light microscope. So here is a little bit of idea for you to understand. So here you have the centimeters, and here you have nanometers, millimeters, and micrometers, right? What you can see. Usually you can see up to here 1 millimeters. That's what you can see maximum, right? Anything below 1 millimeter, it's impossible. What do you have to do? You have to use a light microscope. So the light microscope is where you see the plant, the animal, and some bacteria. So up to the micrometers, the light microscope works. But then after micrometers, when you go even more smaller to the nanometers, the light microscope will not work. So that's where you use the electron microscope. And this is where you see virus, the other internal pieces inside the cell, atoms, other proteins and stuff like this. These are what you find them in the electron microscope. This is the reason why if you decide to make a lab report related to microscope, remember, they don't see every single thing inside of your cell. So you got to be very careful with that. What are you using to see what exactly? Now, since you're talking about this, we can also talk about the sizes. You're going from a cell that is the biggest to the organelle smaller than the cell, bacteria is smaller than organelles, virus is smaller than bacteria, membrane is smaller than virus, and of course, molecule is smaller than membranes. So this is just to give you a scope of the sizes. Even better, showing this picture here, which put a human. So obviously, this area right here, as you can even see a frog egg, but afterwards, you can't. You need the light microscope. So these guys right here, the light microscope will work, and these guys will be the electron. So keep in logic, microscope is micrometers. Electron microscope, nanometers. Anything above millimeters, you can see with your own eyes. Keep that in mind. So now let's learn how to use actually the microscope. Right here we have the ocular lens. That's where you put your eyes. Usually here there's lenses that makes it 10 times bigger. That means you zoom in 10 times bigger. That it goes with combination of the oxygen lens right here, which you can see that you can turn around and there's 3 types of pieces, which these are made of lenses too. So these guys can be 20x, 40x, 60x. Now what are these? 20 times bigger, 40 times bigger, 60 times bigger. Then you have the stage and the condensers, which is where you're going to put the slide in order to see it. You have the diaphragm, which is right under the stage where you can turn it and it makes the light come in stronger or lighter because the light source is right under it, bringing the light up. You have the arm to hold basically the microscope. You have the slide holder, which are these little clips here that move like this in order to hold the slide firm. Now, these 2 are the most important ones. Why? Because that's how you're going to find the specimen. Always start with the coarse, which is this big knob right here, the black one. That one is the one that you move around very slowly to find the specimen. And once you find the specimen, then you go to the little tiny knob right here, which is the fine adjustment. But that one, you have to go very tiny slow because that's where you really will see the details of the specimen. So coarse is to find the specimen. Fine tune is to make sure you fine to the details of that specimen. And this is how you use this microscope. Now that we're talking about looking at specimens, let's talk about the measurements too here. So in the eyepiece, there's this thing called Graticule scale, which again, find the eyepiece right here. And you also have the stage micrometer scales, which is found right here and highlighted for you. So these areas basically is helping you find the measurements of the specimen. Now, how does it work? The stage micrometer scale is usually 0.1 millimeters, or sometimes they just say 100 micrometers. So you can find both ways anywhere. Now, in that stage, you look at the eyepiece and you look at how many ticks can fall in that one stage here scale. So if you notice there, you can actually put 50 ticks inside that 100 micrometers. So what does that mean? If you have 100 micrometers here, and you can add 50 in there, so you divide that, and what will you get? You get 2 micrometers. So what does that mean? Every single one of these ticks, it represents 2 micrometers. And you're like, okay, but what's the point of that? Let me show you. So let's say you have a specimen here looking through the microscope. And you see the eyepiece scale, and you have the stage micrometer right there. Remember, micrometer is again in 100 micrometers, right? So what do you do? This right here, you put it to scale, you look at it, and you see how many ticks falls in there. And you can see from this concept that it shows that there's 20 ticks in there. So there's 20 ticks inside that 100 micrometers. Okay, so if there is basically 20 ticks and 100 micrometers, what does that mean? That means each tick is 5 micrometers. Now, look at the specimen right here. That specimen, how many ticks do you see in that specimen? You actually see about 4 ticks. So looking at those 4 ticks, you basically know that if each tick is 5 micrometers, right, times 4, what does that mean? That means it's 20 micrometers. What is 20 micrometers? It is the size of the specimen that you see in front of there. So that's how it works in order to find the actual image of the specimen. I know that when I'm talking about micrometers and millimeters, some students have a hard time with conversion of units. But you really need to know how to use these conversion of units. So for instance, if you're going from meter to become smaller, millimeters, micrometers, nanometers, you're always gonna be going in a division. If you're going upwards, if you're going from nanometers to, sorry, I made a mistake here. If you're going upwards, that's gonna be division. And if you're going downwards, it's multiplications. That means always by 1,000. You're going from meters to millimeters to micrometers to nanometers. So keep in mind that if you're going from a low case here, low number, to a high number, what do? You divide. When you're going from a high number to a low number, you multiply by 1,000. Some students still have a hard time with this. This is why I usually tell students that you can do something as magnification, as in putting the Microscope idea of all the units. So for example, you have meter, you have decimeter, centimeter, and then you have millimeter, and then you have micrometer, and then you have obviously nanometer. So these are the scales that you have, and you can have that cheat sheet in your test. So for instance, if you have 1 meter right here, usually you have the little dots here. What does that mean? That is just telling you that that's 1 meter. If I want to go to decimeters, so I have 1 meter and I want to go to decimeters. So what do? You put a 0 here and you bring that dot here. Now you have 10 decimeters. If you want to go from 1 meter to centimeters, that dot will come down here. You add, so now it's 100. And if it's 1 meter to go to millimeters, you can see that is 1,000 millimeters, which makes sense from here, as you can see, right? There's 1,000 going on. Then, if you want to go to micrometers, this is where the students make the mistake. They think it's just another 0, but that's not true. The micrometer actually has 3 zeros on it. So in this case, this is how much it will be to get to a micrometer. And if you go to nanometer, it's not an addition of a 0, it's actually an addition of 3. So that will be, again, you put all these 3 plus another 3 plus another 3 in order to get to nanometers. So if you use this table, it will help you so much more in doing the conversions, if you're going upwards or downwards. So for example, if you have 2 centimeters and you want to get to nanometers, you put a 0, 3 zeros plus 3 zeros, and that is conversion from a centimeters to nanometers. But then students will say, well, what if you do the opposite way? Like, how can you go from the bottom to the top with this kind of table? It's very straightforward. Let's say you start with the nanometers 2 here. You still have to add those 2 zeros and the micrometer 2 zeros. And let's say you want to get to millimeters. There you go. You just go back and forth. That helps you a lot if you forget the system. What if you're like, I don't know which way? Like, I made a mistake right now saying, shoot, it's the opposite. You might make mistakes like this. Which way do I divide? Which way I go? If that happens, then use this table. It makes it much easier for you to remember. So now let's talk about staining. When we talk about staining, you have the iodine, which is stained for the plants. You have methylene, which is for the animals, and gram staining, which is for the bacteria. So these stains just make them look much easier in the microscope. So how does it work? First, obviously, you're going to stain the specimen. In this case, is an E. And then when you stain it with a drop, you're going to get cover slip. And that cover slip is preventing the dryness. So the cover slip, if you notice, you hold it in the sides and you let it slide like this. And then it will smear it out and be nice and smooth. All of this just to make sure it doesn't dry, because the dryness will cause the problems with it. So here is the next thing after you have done the specimen, is how to get those magnification numbers. So we know that the eyepiece right here is always 10 times no matter what. That means right here, this guy here will always be 10 times bigger than lenses. The objective medication, which are these guys right here, they are basically saying that you're going to have different magnifications at different powers. So for the low magnetic powers, I would say that usually they can be 10 times bigger. Medium will be about 40 and higher power will be 100. So what does that mean? When you get to the total magnification, you have to multiply these guys. So 10 with 10 will be 100. At 10 with the 40 will be 400. And this will be 1000. That's how you know how big your specimen got. So this will always again be 10. This you have to look and see in the writing what is written, 40x, 45x, 60x. That's telling you how big it is. And that's how you find the total magnification. So now talk about microscope drawings. We have to use a pencil so you can erase in shades and stuff, whatever it is you want to do. Make sure it's in a white paper. Also make sure the specimens are drawn to scale. Like what you see is what you write. When you draw, label them, making sure you also tell exactly what kind of magnification you're using. I'm using the 40x, whatever it is. Label all of them very nicely on the line. Keep a straight line. Do not cross the lines. And of course, make sure that the lines are touching the exact spot. Now, all of this I'm saying, let me show you what it looks like. This is what it looks like, a drawing that you have to do. You can see the nucleus right here. It is touching the right place, the cell membrane, the cell wall, the cytoplasm. They're in the right place. I put the magnification. You don't see me crossing, let's say crossing some lines here. If you're crossing lines, that's a bad thing. Just keep them nice and smooth and straight lines, because that will help you with getting good points. So, done with the drawings. Now let's go to the magnifications. When it comes to magnifications, we are basically talking about how to measure whatever specimens we see in the microscopes, besides the one that I told you about the eyepiece and, of course, the stage rulers. This case is basically when they give you the numbers and you have to find the things. So this equation right here, you really have to know. This will definitely show up in your paper. It will show up. And I will show you examples of what kind of questions will show up, but you have to know this equation really well. Magnification is how big it is. The image is actually the drawing or the picture that you see in the paper. And I mean it. It is the image, the picture that you see in the paper. And the actual specimen is actually the size. You don't see that in your paper or in your task. It's something possible. It's in real life. So it definitely will be hard for you to know unless they give it to you in the numbers. So you're going to be working on these with me right now in some samples of it. Again, so just remember that you always have to keep the conversion units in your mind, because here they're going to trick you a lot. You can also use this table right here that a lot of students they use this in order to be able to remember how to play with the numbers. So for example, magnification is image over the actual size. If you want to isolate A, then it's actually like this, image like this. And if you want to isolate the eye, it's just magnification times A. Now, it's very straightforward. It's a C. If you hear I here with the A, you can see there's a the division here, which gives you this guy. Or you can do the opposite. When it's division here between these two, then you get the A. And then, of course, if you want the I, it's just multiplying those two. So this is how you use this triangle and how you can separate these three ways of using the calculations of magnification. So let's look at the first one right here where it says calculate the magnification of micrograph. If the image size is 300 millimeters, so that means the image right here, it is 300 millimeters. And the actual size of the specimen, which is A, again, you see the word actual size, that is 10 micrometers. Now you see those two, they're not exactly in the same unit. They have to be in the same unit. You cannot make the calculation with them like that. Otherwise, you get points off. Or you don't even get the point. So in magnification, they want to find it. Image is over the actual size. So you have 300 over 10. Some kids go, oh, that's easy. I just can put 30. No, you got it wrong. Because they're not in the same unit. So you have to decide which one you want to convert. You go from millimeters to micrometers or vice versa. So of course, 300 here to get to micrometers, that means I put 300 more, and then I have 10. Now we can cancel these guys, and there you go. You have this magnification. You remember to change the units to always be the same. So now what if you they don't give you the values? In this case, they're not giving you the values. They just gave you a picture, and they gave you some kind of line with a number right on the bottom. What does that mean? And they tell you, go find the magnification. And they're like, miss, something's missing here. No, it's not. The trick is in the picture. Look at this. We want to find the magnification. But what is the image? I said the image, it's the picture. It is the drawing on the paper. So what do you need here? Obviously, what you're going to need is a ruler. So I'm putting the color that you can see. I think let's keep it the green here and make it dark. So the ruler, you go ahead and put the ruler here, and you take the measurement of this guy. And let's say this measurement, the ruler comes in, and let's say that it came out to be in your ruler, I don't know, maybe 10 centimeters. Just exaggerating, but let's say 10 centimeters. So you find that just by putting the ruler on your paper. And then, okay, but how do I find the actual? Look at this. This is telling you that from here to here, it's 2 micrometers. Remember, every image of those tiny things are in micrometers. So you gotta put this guy as many times as you can right here. Let's say this is the size. You put your ruler right here, and then you put the ruler and see how many times this ruler can add here. Okay? So it came out 5 times. I can put my ruler. But of course, put the ruler on the 2 micrometers, see how many ticks it takes it, and then you put how many times you see in there. So in this case, there was 5 of them. If there's 5 in them, what does that mean? It's 2 times the 5. That means it's 10 micrometers. Voila! You actually found the actual image. It's a trick. Learn how to use this in IB. So obviously, the centimeters, I can convert this into micrometers. So you will have magnification as 10 here divided by 10. Obviously cancel, and you have 1000 magnification right there. So very straightforward. Make sure you look at the picture for these ideas. This one right here, again, they have a different value. They actually gave you the X. When they give you a number with the X, that is the magnification. So the magnification here, it is 5000X. You get the ruler, you will put the ruler right here, and you will measure this, and you can say that the image, I'm going to put 10 centimeters again, and then they want to find the actual image. Again, so the magnification is equal to image over the actual size. So if you flip this, it will be A equal to I over M. So in this case, what is the I? It's 10. What is the M? 5000. And then you just divide, and you will find and whatever the answer will be in centimeters. Now here is what I tell students. You got all this. Oh, great. I don't have to do the conversion. Be careful. Sometimes IB will say, give me the actual specimen size in micrometers. So you go through this whole process, right? Because this is in centimeters and there's no unit in the magnification. You get a centimeters. But the question is asking in micrometers. Once you find the answer, convert it to micrometers and then. So be careful, look exactly what unit they're asking for. Because also students make the mistake of doing this whole thing really well, but then forget to do the final conversion of the centimeters to micrometer as the question asks. So you gotta be careful with that. So now, in the next couple of slides, I'm just going to give you exposed to some of the micrographs of pictures. Like this one right here, you can actually recognize that it's a long guy here that usually looks like a rod, which is basically a bacteria. How do I know that? Simply because you can see the darkness in here, which this darkness, it basically means that is nucleoid with all the DNA. Now, the line of the membrane here, you can see that there is a line right here, which is inside, that's the cell membrane. So this is one spot of a bacteria. Now, right here, what exactly do we see? We actually see a plate. But you're going to say, but it doesn't look like a square. It doesn't have to. When you see something very large like this, that it's white, that's a vacuole. And then when you see a large circle, it doesn't have to be a perfect circle, it's a little bit more darker, that will be the nucleus. And then you have the cell wall right here, the cell membrane is inside, and then you can see the mitochondria, and you have the chloroplast, so you can easily identify this as a plant. It is easy for me because I've seen it many times, so it's important that you keep seeing as many as you can. This one here, now we can see that there is this big, huge, dark nucleus here. You can see the mitochondria, which you will see a little bit of a straight lines, dark lines in there. You have the plasma membrane, which is the outer part. You got the centrioles, and you have the Golgi right here. But this, you can easily be spot that it's going to be, because you don't see a large vacuole. You don't see this huge wide circle in the middle, so it definitely cannot be a plant. Right here, again, you see the white big spot here? That is a vacuole. And then you have this huge thing, which I'm trying to show you not to get the concept of this, of nucleus always being a circle. You can see it's not really a circle, but it's a long one that could look like a circle. Because again, we make it nice and prettier when we do the drawings, right? You can see the mitochondria here. You see the chloroplasts. These are chloroplasts. You'll see like the dark lines, and it'd be like almost like a dome and so forth. And you can see the cell wall will always be the outer line, and the plasma will be the inner line, just to keep you in mind. Here is an example of a mitochondria. You can see that because you have these lines like this going in, and it looks like a rod shape too. Why is this not a bacteria? Because you don't see a dark in the middle here, okay? Then you have here the chloroplast. Chloroplast, when you see a lot of these stacks of blackness in there, that is the thylakoid. So that's part of the chloroplast right there, okay? Now here we have the Golgi, and you can see the trans and the cis phase. The Golgi is, like I said, because Golgi usually looks like this, right? And you will see little circles going on. So you can see these little circles right here. I'm going to put another color. You can see these little circles right here, and you can see that it's all of these guys going on. So it's very easy to spot. Remember, if they want you to identify, they will not zoom out and, like, for example, let me go back here so you can see in the animal part. They will not put the zoom out like this and tell me the Golgi, but you see the Golgi here barely can be seen. They're not going to do it. I mean, IB can be such a pain sometimes, but not that level. When they do that, they definitely going to zoom in and tell you, hey, what is this? So they're going to zoom into the pictures and make it much easier for you to identify that it's a Golgi, okay? Here on the rough one and the smooth one. The rough one, you can always see that there's going to be dots in there. So it's going to be a lot of lines, as you can see, with dots. That's the rough. When you see lines here, a bunch of these circles and lines going, like small lines, but a bunch of circles, that will be the smooth ER, because there's no dots, because there's no ribosomes. So keep that in mind. Then right here, identifying, remember the cell wall is always the outside, the plasma membrane is on the inside. That's the most important thing. So make sure when you touch the line, like what if I touch here, that touching line on the outermost line, that will be the cell wall. And then when you go to a little bit lighter one, that will be the cell membrane. So keep that. Right here, we have the chromosomes and the villi. How do I know this is a chromosome? Because chromosomes, usually it's an X, and you will learn more about them once you talk about mitosis and meiosis, how they condense into an X. So, and then here is the centriole that connects those two chromatids. Here, what you see these big lines here, that's the intestine. That's basically the villi that you have. And here you might have some of the mitochondria going on. And then right here, we have basically the pancreas cells. We have secretory vessels right here, all of them catched up all together as one. So you have the pancreatic, and here you can see that it's a bigger one. It's more zoom in about these little circles. So this is it for this section. So what do we learn? We learned about the microscope parts, about magnification, how to use the magnification's lenses in order to measure things. We talk about different types of microscopes. We look at some structures of cells and be able to label them. We also talked about the magnification equation, how to use it in different situations. Also, we talk about different types of microscopes and their application, and that's pretty much it for this video. This is a scope for it. Now, we are done with A2.2 for the S cells. If you're HL, go ahead, you go to the HL section of it. If not, then for the S cells, you go into the next one, which probably will be diversity of organism. and I'll see you then. Remember, with NAIB, you can get that 7. See you soon.